Good day, this is the Professor, CBMD PhD, and today's Monday, November 20th, 2023. It is 2:15 and 17 seconds a.m. Eastern Standard Time, and today's research, lung cancer. So let's jump to it here. As gene splicing reduces effectiveness of CD20 targeting monoclonal antibodies designed to treat a variety of blood cancers and disorders, researchers found CAR T-cell therapy may serve as an effective alternative for patients with these disorders. Date December 16, 2023. Source: Children's Hospital of Philadelphia. Immunotherapies that target the CD20 antigen have revolutionized how patients with a variety of blood cancers and hematologic disorders have been treated. However, many patients develop resistance to these treatments due to a loss of the antigen that is being targeted. Now, a new study has found that gene splicing occurring within these cells can cause significant changes in CD20 protein levels that render the therapies ineffective. The CD20 is a cell surface protein involved in the fine-tuning of B-cell responses to foreign agents like viruses. It is expressed exclusively on the surface of normal and malignant B cells and is not expressed by other cell types in the body. And this makes CD20 an attractive target for monoclonal antibody therapies, which have been used to treat a variety of B-cell lymphomas, including follicular lymphoma, Burkitt lymphoma, diffuse large B-cell lymphomas, and high-grade B-cell lymphomas. And despite anti-CD20 therapies revolutionizing the way these conditions are treated, some patients with these conditions do not respond to antibodies targeting CD20, while others initially respond before eventually developing resistance. As prior research has shown that the loss of CD20 reduced the effectiveness of these immunotherapies since it would remove their intended target. However, the mechanism by which CD20 levels can be reduced were poorly understood. The researchers at CHOP suspected that CD20 messenger RNA was not being properly translated into the CD20 protein expressed on the surface of cells. In this study, they found that certain isoforms of mRNA responsible for producing CD20 were impacted by splicing in a way that the proteins were not being made at the levels necessary for these immunotherapies to do their job. In the study, researchers focused on the MS4A1 gene, which encodes for CD20. The gene undergoes splicing or stitching together of its building blocks, called exons, to produce several mRNA isoforms, which may encode the identical amino acid sequence, but differ in the efficiency with which the protein is made. The researchers identified 4 variants in total among normal and malignant cells. Of the 4 variants, V1 and V3 were far the most abundant, and only V3 is efficiently translated into CD20. In contrast, variant 1 had trouble recruiting ribosomes responsible for making proteins, making it difficult for monoclonal antibodies to target affected cells. They also found that CAR T cells were still able to effectively kill both V3 and V1 expressing cells. CAR T cell therapy modifies patients' own immune cells to kill cancer cells. As historically CAR T cell therapy presented challenges, it was thought not to be effective in these malignancies, but the study showed that it was more effective in targeting both variants compared with the monoclonal antibody mosunetuzumab, which is only effective against V3 expressing cells. If a patient has relapsed because CD20 levels are downregulated, CAR T cell therapy may still be an option, as it requires a lower threshold of the protein in order to be effective. These findings may help clinical staff offer more precise options when treating these hematologic malignancies. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.